It's our last talk of our crowdsourcing theme quarter, and boy, we have a great one to end with. This is Adrien Treuille. He's a VP of Simulation at Zoox, which is one of these self-driving car companies that has raised a lot of money. I think they raised something like $250 million in Series A funding this year, doing some really interesting stuff. Adrien, I interacted with Adrien because he was a professor at Carnegie Mellon in CS and robotics before he got sort of pulled away to work on a Google X project, which we know very little about. But, so Adrien has won numerous prestigious awards, including the NSF Career Award, an Okawa grant, and the MIT TR35, which is an award for innovators under the age of 35. And much of the work he's sort of received these accolades for are on the two projects that he's known for, EteRNA and Foldit. So both of these are citizen science games that have attracted hundreds of thousands of people to play, to do real science work. And he's going to tell us a bit about that today. And welcome. Thank you.
Thank you. Yeah, Stephen, first of all, thanks for a really wonderful visit. I love this campus. I love how engaged all the students are. It's just been a great day. So thanks for inviting me. And actually, to that end, I would just be totally delighted if someone raised their hand and said something in the middle of my talk. No one ever takes me up on that offer, but you can be the first. So yeah, I'm going to talk about crowd solving, and which is a little neologism based on the familiar crowdsourcing. And I guess the reason why I wanted to use this word is that often when people think about crowdsourcing, they think about simple image and pattern recognition tasks, which we then want to do, which computers are not as good as humans is doing, and which we want to do on a very large scale. And then some people additionally also think, well, if we can get people to do these individual computational tasks, and we kind of assemble the people into a larger computational unit. I think that this approach has proved to be very important and exciting, but there's a lot more that we can do at the intersection of large-scale human involvement and computer science and HCI. And I think that really, first of all, we can solve extremely complex problems. So we don't just need to do things that humans can do reflexively well. We can use the crowd to solve really difficult problems. We can also identify people who are actually particularly good at solving problems. So we don't need to view the crowd as sort of a homogeneous mass of people. There might be some prodigies in the crowd. How can we use that in interesting ways? And finally, we can extract large data sets which are essentially we cannot extract in any other way. So I'll talk about that at the very end. So my hope throughout all the projects I talk about is to inspire you to think that really fundamentally new approaches exist and are still possible and are still discoverable in crowdsourcing. And therefore, I like this term crowd solving, which is really trying to tackle very, very complex problems in the context of large groups of people and computers. And I'm going to do so in the context of asking a series of questions about crowdsourcing. In the interest of time, I have omitted some of those questions. So if you notice it going from question 11 to question 13, you're not going crazy. I just omitted question 12. We can talk about it if there's time at the end. And I'm going to talk about these three projects. So the first one is a game for protein folding. The second one is a game for Rna Engineering. And the final one is for understanding perception in the context of visual depiction. So let's start with protein folding. How many of you know what protein folding is? The protein folding problem. Okay, cool. Yeah, UCSD has a good biochemistry. So proteins are these incredibly beautiful molecular machines that basically carry out most of the basic functions of life in the cell. And they fold, they come out as these strands of amino acids, and then they fold into these really like hallucinatorily beautiful shapes. And they're even more beautiful when you color them rainbow like that, which has nothing to do with reality, but it's actually quite interesting. The rainbow, you color it, this is one coloring scheme, you color it basically just starting red, orange, yellow, green, blue, purple. And when you do it this way, you can actually realize the incredible sort of redoubling that the protein does on itself as it folds. These are really, really complex shapes. Of course, these are all real proteins. And this is an animation of what it looks like for a protein to fold. I guess the intuition here is that when you're at this molecular level, the world just is not really at all like the sort of world of solid forms that we are familiar with. And what's actually happening is that everything is vibrating and jiggling. So I was talking to someone about computation using vibrations. Well, anyway, this is an example of that in some ways. And then sometimes some of those shapes, or conformations as they're called, turn out to be more energetically beneficial. And when the protein finds one of those, it's more likely to remain in something in that shape or in something resembling that shape. So this is the process by which everything sort of bouncing off each other, the protein essentially forms a particular shape. Okay, so the way that this is modeled computationally is that the protein is regarded as a series of atoms, essentially, and really you can't pull on the bonds because that's just extremely energetically unfavorable. But what you can do is you can rotate along them, and that's okay. The protein doesn't mind that, and that's actually where the flexibility comes from. But sometimes when the protein does rotate along its bonds, various atoms will become in contact with one another or closer to one another, and then this whole myriad of forces essentially determine how good it is. So if you view these as theta and phi, so basically we have this energy function, and just computationally it takes all the theta and phis and it gives you a number, and that's how good your protein is, or I should say that's how well folded your protein is according to our understanding of protein dynamics. And that's an important caveat since our understanding is incomplete. But there's a lot of these energy terms here. So there's Leonard-Jones potential, which is sort of the general propensity for atoms to get close to one another. There's hydrogen bonds, there's solvation. Nowadays, a big deal is just statistical energy terms, so they're not based on physics, but they're based on sequencing or imaging tons of proteins and then figuring out statistically how does it fold and adding that into the energy function. And then it turns out that if you regard this as sort of an energy minimization problem, the energy landscape is just incredibly hilly and forbidding. So you can be very close to the right solution and have very bad energy, as it were. And in some cases, you can be very far from the right solution, and actually the energy looks pretty good. So it's quite difficult to do this. And actually, a sort of amazing fact is that in the body, a protein can take minutes to fold. It's really remarkable that there's incredibly fast vibration. I mean, huge amounts of computation are happening, as it were, in the body. So protein scientists, biochemists, and biophysicists have really gone to CLR. They've gone to the algorithms book, and they've used every possible trick in order to optimize the energy of proteins, and it's still quite a difficult problem. And so we asked a rather unusual question, which is, given this famous multi-decade-long science and computer science problem, could we turn it into a computer game? And the intuition here is that while there are many things going on when a protein folds, really a forbidding number of things are interacting with one another. As it turns out, if you ask a biochemistry grad student who works in structural biology, is this protein, does it seem like it's well folded? Or does it seem like the algorithm got messed up? Often they can tell you. So, and they're actually the ones writing these algorithms. They have to look at these things all day long, and they know when their algorithms aren't working. So they're able to actually create a sort of intuition for what a well-folded protein looks like. And if that's possible, then maybe we can tap into that intuition. We can train a lot of people to have that intuition, and then we can use that to accelerate protein folding. So how do we turn protein folding into a game? Well, the first thing that we had to do was actually come up with visual metaphors for all of these different forces that determine how proteins fold. And this was one of the most fun parts of creating Foldit for me. So here, these two side chains, they're called, little groups of atoms, are too close to one another. And so they're repulsing one another. And we played with so many variations on this. Should it be a gradient of how red they are based on how much they dislike being close to one another, which is actually physically how it is? Or should we abstract it, which is what we ended up doing for human cognitive load, so that it just shows just turns on into a red color when it's past some threshold. We created how do you show when a protein is, how do you show attractive forces? Actually, that's really quite difficult. And in this case, the way we do it is by basically putting some little bubbles inside the protein. And these bubbles have no physical meaning. They indicate space. But when the protein gets more closely folded, the bubbles pop. And it's really satisfying. And then there's other bonds which are showed with various color schemes and stuff. So,
Hey, Adrien.
Yeah. Oh, thank you.
I have two totally different questions. One is, in 2016, can we just take a picture of things? How come you need algorithms to know how they're folded?
Right. So that's a great question. And the answer is that in 2000, well, the answer is that there are many people better qualified to answer this question than me, but since I'm the one on stage, I'll do what I can. The basically high throughput synthesis and imaging of proteins has gone through like a revolution, actually, since we published Foldit. And so in many respects, some of the emphasis on computation is now gone. So you actually can just be like, let's create tons and tons of variants of this protein, and then let's image all of them, and let's see how they work. Whereas when I started, I mean, getting your protein synthesized and imaged was like something that happened like once in your PhD or twice in your PhD. And by the time we were finishing Foldit, it was something that happened like once a month in your PhD. So already like the revolution was underway. And I mean, this really exciting field actually. So yes, it's true that the sort of overwhelming need for these incredibly sophisticated computational methods is less than it was 5 or 10 years ago. On the other hand, the interest in protein folding is not only scientific, in other words, how do they fold, but also design. So if we wanted proteins that folded into a particular shape or variants of proteins, how would we design them? And that's something where still you would like to have computer assistance in order to be able to go through like large numbers of designs before you start actually synthesizing things. So yeah.
And my second question is totally different, which is how did you stumble into this problem? Like this project has just been so insanely successful. Did you discard 99 other domains, or were you just always fascinated by proteins? Or?
Right. No. So it was the opposite. In the sense that it was really specifically protein folding. And so what happened was the story is basically this lab which had been doing protein folding at a very high level. They'd been winning the protein folding competitions for many targets for many years. Someone had a few years before I had joined created like a little thing where you could move two proteins and get them close to one another. And this meme was formed that actually it was sort of like a game. And you can actually watch some old videos of people from the lab saying like, oh, it's sort of like a game. Then side note, the lab tried to con me into joining them to do GPU programming, which I had no interest in doing. And they utterly failed to recruit me, but I sort of heard about this meme and I was like, this is what I want to do. And I basically that week canceled my postdoc at a prestigious other university and canceled my life basically and started Foldit. So yeah, that's the real story. Yeah. Yeah. So okay, so and then there's a lot of right. So then there's a lot of interactive. Basically interaction modalities which had to be invented in the case of how you can fold proteins. So you can pull on it in various ways, but you can also pull on it having locked various pieces of the protein. And then, really importantly, you can ask the computer to optimize the protein. So it's not as though we threw out the previous 3 decades of computational optimization in proteins. Instead, we basically just co-opted them and brought them into the game and made them buttons that you could press. And so wiggle basically means that the protein energy is continuously optimized. Shake means that you do sort of a Monte Carlo optimization over some subset of the protein. And then rebuild and tweak are other variants on discrete and continuous optimization. So the computers were very much in the loop, even as players were playing the game. Okay. And then of course, we spent a lot of time working on tutorials and how do you educate the user in-game to get them to understand protein folding. And finally, a huge aspect of Foldit was the forums. And so in fact, there's still a basically untapped giant mass of data, which is all the communication among the players for now, it's been about 10 years, as they try to work with one another to understand how to fold proteins. And this is what the game looks like. This is my nephew, actually my cousin, playing Foldit in upstate New York. And yeah, he was one of, sorry for the promotional aspect of this video, but he was one of hundreds of thousands of players who were folding proteins at the same time. So not only was this just really, really fun to put together, but as it turns out, there were we actually were able to demonstrate that humans were better than computers at folding proteins. And so in this case, here you see this. Okay, so here we see three proteins superposed. Now the blue is the native conformation, so that's the shape into which the protein should fold. The red is the best computer solution, so that is the best thing that the computer was able to come up with, and the cyan is the best player solution. And the key point to notice, actually interestingly, is first of all, they're very similar to one another at a macroscopic level, but it turns out that the computer kept this loop, it's called, pointed out. Whereas in fact, the humans were able to figure out a way of pointing it in. And in fact, that's what nature does too. So, so for all the computational muscle which we were able to throw at the problem, it wasn't able to sample the correct solution. And so this culminated in a really fun paper in 6 years ago where we demonstrated this result on a number of blind targets. So it was really exciting. Another really, really amazing thing for me, basically life-changing about working on Foldit, was I had worked on a lot of sort of dry academic problems, or I should say fun but very mathematical problems. and you know, my users were like the program committee of the people who read my papers and commented on them. And suddenly I had real users. And these people just came in, and it was like humanity just streaming in the door, and it was really blew us away. And I mean, there's so many stories about the players, but here's one. Nature actually did a documentary on some of the Foldit players, and this woman was at the time the #1 Foldit player in the world. And it turns out that she was, well, you'll see.
I'm administered working in the rehab team. I'm just answering telephones, working on bespoke computer programs, interacting with staff. It's very repetitive. When I go home, I become a different person. I just like to measure myself against something, and it's given me something that my everyday life hasn't given me. Is to just use abilities I didn't know I had with a protein that is so misshapen, it's ugly and it's red, and to work through that to a beautifully streamlined structure where it's symmetrical and things are tucked in and nothing is hanging out of the sides. Yeah, that is sort of the goal, and that's what I like doing, to sort of help it in some ways to sort of be what it should be.
Yeah, so I guess in conclusion, I think an important thing, really the most important thing sort of computationally for me about Foldit is that a lot of people thought, oh, this is humans against computers. And in fact, when we were first presenting this project, especially to biochemistry audiences, there was like outright hostility. I remember standing in a stage on an audience about this big, and this really eminent biochemist was like, this is total BS, and there's way too much going on, and there's too many forces, and you're not going to be able to get anyone to do this, and this is a waste of time. And perhaps, that's very intimidating to hear when you're a postdoc. But perhaps actually something that even I didn't really fully recognize at the time is that it's humans and computers against computers. And so we were really able to co-opt, and sometimes we were doomed to succeed because all of the human computation was coming on top of the existing computation. In fact, it was not just humans versus computers, it was networks of humans working together versus computers. And then in the context of this very elaborate visual interface and the forums and everything. So this was also for me, a view that really fundamentally new techniques do exist in crowdsourcing. We were also able to identify people with special skills, really amazing Foldit players, and I'll talk about that in the next section. And finally, that we're able to solve these extremely complex problems. So this was not just, where are the faces in this image? This was protein folding. So that was really exciting. And so armed with these insights, we decided to tackle another really exciting and very unique problem, Rna Engineering. Any questions about Foldit? Yeah, the person behind Scott?
It's still going on. It's one of the 15 years. So
Yeah. It is still going on. And it's sort of an institution now, actually. Like, people come into the lab and they add a feature to Foldit and stuff. And the game has evolved a lot since its original form. So. Yeah, yes,
okay,
you can.
I'm sorry.
Yeah, you absolutely can. Scott.
And how much did you advertise it? How did you build up a community?
Right. Well, I'm prepared to answer this question because Stephen asked me the very same question this morning. So we really played a very good trick, which I recommend you play if you can, which is that we got the mainstream press to write about it. And we had a connection, as they say. And we basically, we played all the right tricks, which was that we had a story under embargo, and the story was going to come out the same day the thing launched and stuff, so the reporters were really excited about that. And we managed to get a story in The Economist, and then that led to sort of all these follow-on stories and stuff, and that really put a lot of people in the door on the first day. So that it would not, I'm not actually sure what would have happened without it, but I think it would be quite difficult to get it up and running. Because the community needs to be there, basically, on day one. Our community before we launched was probably about 100 grad students or something like that. Okay. So yeah, the power of the mainstream press. Okay, so Rna Engineering. Well, we were talking about the power of humans and computers versus just using computers to solve scientific problems. And I'm going to talk about it in a new domain now. But there is another really exciting element to this game, which is that we're going to add actual experiments in the loop of the game, so that we're actually going to be synthesizing molecules and measuring how well they work. And that's going to be part of the scoring mechanism of the game. So, okay, RNA. So RNA, the story used to be, well, RNA, remember, I don't know if you remember this, but there's DNA, and then it sort of becomes RNA, and then that sort of becomes proteins, and then you get an A on your 5th grade science quiz. And that's actually what everyone thought. But it actually now turns out that RNAs fold into extremely complicated molecular machines themselves. And in fact, the machine that builds proteins, which is like the most complicated machine in your cell, is in fact mostly made of RNA. And so there's this hypothesis now called the Rna World Hypothesis that really the earliest form of life was RNA. And then RNA invented proteins to do its bidding. And then it invented a DNA to store information about how to make proteins. And then it invented all these other things around itself. And then when we finally looked at it, it was like the master. It was so much the master that we didn't even realize its mastery when we first discovered it. But anyway, so this is an RNA molecule. Actually, this is that generates proteins, and it's similar to the folding problem in some respects. We would like to be able to design RNAs to create next generation catalysts. RNAs actually change shape very quickly in the cell, which seems to be one of the keys to their usefulness. So we can create drug responsive control elements. But because they were much, much less well understood than proteins, their folding models were not very well understood as well. And that means that there is this exciting opportunity to not only study how they fold, but to actually try to figure out the principles, the rules of the game itself. Okay? So, how can we take all of these elements of the field and generate a game in which we try to figure out how RNAs fold, and in particular, based on a limited number of weekly experiments. So at the time, we figured out that given our budget constraints, we could synthesize 8 RNAs every 2 weeks. And we had to sustain a population of, we were hoping, hundreds of thousands of players on that tiny little trickle of experimental data. And so what we did was we created this really fun, cool interface you see here. You can also download this one. Yes. Actually, we made a bad bet on this one, which we wrote it in Flash. Which nowadays, if you want to use Flash, you have to click yes 100,000 times. It's really annoying, but it's worth it. Anyway, so we create this beautiful design interface where players can design RNA molecules, and then every week they would vote on which of their player-submitted designs were considered the most likely to succeed. We took the top 8 votes and we actually synthesized those in the lab. And then we sent back the results in the game, sort of in a seamless interface, so that the players could see how well they were doing and compete with one another. So if you think about this, we actually have everything from hypothesis to experiment to result. We're sort of crowdsourcing the entire scientific method. And so this game also made a splash when it came out, and we were able to garner a lot of press, and that was really important in sort of building the community. And then a couple of years ago, we were able to publish our first paper. And at the time, so we had about 75,000 registered players who'd contributed 300,000 man-hours, and we'd synthesized 40 DNAs. Or 400 DNAs, I should say, which is on the order of 10,000 individual data readouts, and the results were really stunning. So here's actually what we're trying to do in the RNA game. We give them a diagram of what the RNA should look like. And this is sort of a 2D diagram. In reality, all of these things would actually be helices, and there could be some additional 3D structure. But this actually is a very informative diagram. It basically says that this amino acid this nucleotide is bonded to this nucleotide in a Watson-Crick pair, and so on and so forth. So you can think of this as like an engineering blueprint, a 2D engineering blueprint of a 3D RNA. And we would say, we want you to come up with a sequence of nucleotides, A, G, C, or U. We want you assign one letter to each one of these dots, such that if we synthesize this, it'll actually form this shape, or something close to it. And we're not going to tell you the rules of RNA folding. This is what we're going to give you. And here are some of the player designs. And you can see that they actually have that 5-pointed starred shape. And they have these wonderful names like Starry Jess's star is called Starry's bulge star. And then these were the designs created by the computer. This was the state-of-the-art algorithm at the time. Okay, so it wasn't actually always this easy. So when we started the game, actually the computers, the humans were doing pretty horribly. So this is a plot which shows you what the humans and computers were doing. So each one of these dots is a synthesized RNA, and then the higher they are on the scale, the better they are.
What is a square and what is a circle?
I was just asking myself that as I looked at this diagram. Yeah, someone should find that out. I'm sure it's in the paper, but I must admit that I forgot why there's squares and circles. But anyway, and I'm actually there's a mistake in this slide too, I think, which is that I'm pretty sure that the red ones are actually the computer and the blue ones are the humans. And it's mislabeled here. But it doesn't make a difference. The point is that they're actually both doing pretty badly. So they're all over the place. And as it turns out, for all of the work that had gone into RNA folding, if you actually took the state-of-the-art algorithms and synthesized the RNAs that they told you would fold into the correct shape, often it wouldn't work. And it turns out if you asked a bunch of random people on the internet to do the same thing, it wouldn't really work either. And that's what you're seeing right now. But the humans had access to this really amazing thing that the computers didn't, which was that they could talk with one another, and they could design new RNAs, and every 2 weeks, like clockwork, they would get this new stream of empirical data telling them, this is how your RNA worked, this is how it didn't work, and it was really very rich. It was actually per nucleotide data we could give them. Did it bond properly? Yeah. And so what we saw over the first 6 months, 6 to 9 months, was the humans were getting better and better. And the computer actually was getting worse and worse over time until in about 6 months, the worst human design was better than the best computer design. Yeah?
So the humans had access to feedback, their experiment data.
Yeah.
Did the computer algorithm have access to this sort of as a training set so it could continue to improve, or?
That's a really good question. So the computer did not. And in a sense, that's just not how it worked at that time. You would download a computer algorithm and it would have a lot of magic numbers in it that had been estimated at some point in time, and then you would use it to synthesize, to design an RNA. As it so happens in a slightly different way than you're describing, the numbers have been updated since then. And so the algorithms that we're comparing against now have like new energy parameters in them which make them fold better. So there is a moving target between the state-of-the-art computer algorithms and what the humans can do. But at this point, we were comparing against the state-of-the-art algorithms as they were understood by science in 2013. And then our players were doing what they could to understand things. Yeah. Any other questions? Yeah.
So along with time, is the difficulty of problems also changing?
Yeah. Yes, that's a great question, and that actually explains. So, this is unfortunately not shown in this diagram, but in fact, what's happening here is that we're actually changing the target over time, and we're making it harder and harder, and that explains why the computer actually does worse, in a sense. After 6 months, when the humans do better. And in fact, if you do this over a longer period of time, it's very dramatic that the computers are doing worse and worse and worse, and the humans are doing better and better and better. So actually, ISO difficulty, this graph is even more dramatic. Okay, so Next question is, okay, so we believe that the players have learned something. Is there, that's sort of interesting in and of itself, but it's kind of annoying from a scientific perspective if you're trying to actually do something and you're like, okay, well, let's call up those players on the internet and have them, vote on what the right design should be, right? It would be nice if they really had learned something that we, scientists, could figure out what they'd learned, and that we could potentially even apply it without having them be in the loop. And so the question is, okay, so now you have 75,000 people playing your game, and they're doing better, it seems. What are they, well, not just it seems, actually, I should say. We did all these p-value tests and all this fancy stuff, and they really did better statistically. How can we learn what the players are doing? And in particular, we don't want to learn some like incredibly esoteric deep net or something with 100 million parameters, with all due respect for deep nets. We'd like to learn something that has a little bit more the flavor of science, which is to say explanations for why proteins fold properly, which are parsimonious, which you could just understand, which are empirically verifiable, and which are human understandable. Okay, so. And as it turns out, the players were really aware of this. So I remember talking with Eli Fisker, who's one of our top players in the world, really amazing person actually, and he says I felt a bit sorry. I was chatting with him on the player chat. I said I was feeling a bit sorry for them. They made all sorts of funny errors that we quickly outgrew. And then he actually, it's very interesting, he then tells you in somewhat sort of technical jargon what one of the errors was that he saw the computers doing. So this is really this amazing thing that humans instantly were like, okay, we did something wrong, let's do something differently. And the computer algorithms are just sort of plodding along, doing the same thing all along. So but how do you do this? How do you learn what the players are doing? This is, I think, one of the neatest tricks of this research, and it was actually the biochemists who came up with it. I was thinking very complicated. Oh, let's create a giant Markov model of what the players are doing with 1 billion states, and we're going to estimate everything, and it's going to be like our player model, and of course I was just using my computer brain, and then Riju, the biochemist, said, well, why don't we just ask the players what they're doing? Yeah. It's an interesting idea. So what we did was we created this thing called the strategy market. And so what you could do is write your strategy, see an example post, okay? The EteRNA developers will write an algorithm based on your post. So basically we allowed them to tweet their strategies. There was like a character limit. And they would write down, well, I think that, 3 nucleotides down from a GC pair, there should be like a something or other with this probability. And we had a bunch of undergrads and grad students code up their strategies. And they each turned into maybe a 100-line script, really short, even less, 25 lines. And then we were able to run those scripts against all of the proteins we'd synthesized, and there was a strategy market, as we called it. You could compete over what your strategy was. And some of these strategies were really amazing. And so this is actually one of the strategies which turned out to be extremely empirically predictive of how well RNAs fold. It was discovered by the aforementioned famous Eli Fisker. And it was unknown to science. And it's a very simple observation. He basically says, if you look at this thing called a multi-loop, you read it actually either in clockwise or counterclockwise order. When you see GC pairs, they should always go GC, 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 or else CG, 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 CG. In other words, they shouldn't have GC, CG, GC. Okay, there shouldn't be reversals as you read it in this particular order. This no energy model in any algorithm said anything about this. This was like a totally non-local interaction between nucleotides that no one had thought of. And yet, when we ran it against our database of algorithms, it turned out to be extremely discriminative. It's really amazing. And so what we did was we took all of the strategies and we took our synthesis database, which was growing weekly, bi-weekly, and we ran this really nice algorithm called Ask Lasso, which not only figures out how to combine the best strategies, but also how to throw out all but the very best strategies. And the result was something which we called the Ensemble Strategy, which was just basically 5 rules. Each rule was extremely simple. We could list them in 1 sentence each, and which together were extremely predictive of how well RNAs fold. It's just amazing. So 75,000 players boiled down to 1 validated algorithm. And as it turns out, so here's a result from the paper. So the previous algorithms were in gray and light gray. Anyway, the point is this. The players are in blue and they're the best. And our ensemble algorithm is in purple. And it does almost as well as the players. Not quite as well, but almost as well as the players. So we managed to actually in some ways distill out of them some of the insights. Now, not all of the insights, which was another really fascinating thing. There was something else that they were doing better than what we had figured out. But we were able to get some of it out, maybe 75% of it out. And then, yes?
So for example, the GC thing,
Yeah.
did you end up chatting to those people, or did they explain why they thought it was a good idea?
Really interesting question. They don't know why it's a good idea. And in fact, we ended up getting this published in a very nice journal, but PNAS, but it was first rejected, and one of the objections was that we had not given a mechanistic explanation for the rules. And then we pointed out that the existing rules that RNA scientists use also lacked mechanistic explanation, which is also true. But anyway, no. So that in fact we didn't have in general, and for that matter, the vast majority of the hypotheses were wrong. Almost all of them were wrong. But we were able to extract a very, very tiny number of them, and some of them also, by the way, that were correct were known. That's another interesting thing. So we basically rediscovered principles that RNA sort of scientists had figured out. But in I think 2 cases, they were unknown. And that was not by us, by Lasso, by some algorithms selected to be in the golden set. And in fact, when we tested this on many RNAs, which were completely out of the set of RNAs that we had trained on, obviously, it turned out that it really was predictive. So, yeah.
Were there certain strategies that people submitted that were hard to turn into code?
I mean, you'd have to talk to the undergrads who did this. I was probably the least aware of how hard it was to actually accomplish this task. As I recall, it was a negotiation between the players and the coders, as it were.
I just
So,
wonder if there's something like make it look elegant.
right. So if that were the case. Right, so I actually don't remember the social rules of the strategy market. We did give them an example, which I think sort of gave them a guideline for how to do it. I think it had to ultimately be turnable, algorithmicable, yeah. Okay, I'm going to skip this question, although I think it's really interesting because we're running out of time. Long story short, we have a bunch of people and we know how good their designs are, and we also have a huge amount of information over who's influencing whom. And I think that we should be able to use that information to figure out not only scores, but also perhaps a payment scheme. But I don't want to go into that if we don't have time. So, okay, and I'm not going to go into this one too, because we're really running out of time. So I would say, to Scott's earlier question, we're now in a different world across all of biochemistry, and it's a world that's continuously changing, and they call it super Moore's law. So now we can actually synthesize 10,000 RNAs per week, and that's just a lot more than 8 every 2 weeks. And so I'll let you guys do the math. And so, so long story short, we want to move to a model where the players are actually doing more independent research, and so they are basically allowed to put forward hypotheses, and rather than voting on the designs, which is no longer even an issue anymore, we've got enough designs, or we've got enough synthesis slots, I should say. We want them to vote on which hypotheses are worth exploring, and we're going to sort of like as PI status give them a certain number of RNAs they can synthesize per week. Like you get to synthesize 2,000 per week, you get to synthesize 500 per week, and then allow them to write these things which we're calling tiny papers, which are basically little sort of one-page mini explorations of something that they discovered while designing RNAs. It turns out that first of all, the sort of new models like PLOS1 are giving us new ways to publish scientific data and archive and stuff. So that's sort of an interesting opportunity. And second of all, the community really has a ton of, you might call it like sort of kitchen knowledge about how to design RNAs, which is basically unpublished. So it'd be nice to get that out there in a way where a postdoc at UCSD who's actually interested in some sort of tiny and esoteric branch of RNA folding could actually Google and find that some EteRNA player had actually thought about this a little bit and had some data. So that would be really cool. Okay. So in EteRNA, we extended crowdsourcing to experimental science, which is a really exciting new development. And in particular, the rules of the game, in other words, which designs were going to fold properly, weren't known a priori. And the players actually were able to come up with simple rules to explain the RNA data. And on top of that, we were able to create a new RNA design algorithm that distilled those rules. And then finally, the part that I glossed over is that we can perhaps use some of the information in these games to generate a sort of mathematical understanding of how they're collaborating and figuring out the answers. And finally, I think that we're creating a new generation of researchers attacking this problem, and lots of fun stories there. EteRNA is now used in schools. Sort of NOVA took over a branch of it and created like an educational system around it. So lots of exciting stuff there. Okay, in the last 10 minutes, we're gonna go through understanding perception, right? That should take about 10 minutes. So I guess this is another cool thing that you can do with crowdsourcing. And it is to basically extract data that you couldn't get otherwise. So in the case of, we're in this era of big data, and there's all this amazing stuff that you can do when you have this data, but what do you do when you don't actually, when you can't actually find data that you would otherwise like to machine learn? And this is something that I think we can use games to solve. So to give you an example, here we have an early cave drawing, 40,000 years old, so about halfway to the essentially birth of hominids, or birth of modern humans, I should say, which is about 100,000 years ago. And yet it's a completely recognizable drawing, right? So, and of course, a computer would not recognize this as a bull, but all of us would instantly. So there's something about human perception that's encoded in this drawing that's really interesting, and that we'd like to sort of get more information about. And so similarly, here we have a picture of one of my collaborators, Michael Cohen, wonderful man, and a bunch of drawings that were generated as part of this project. And they're really wildly different, and yet they're all clearly of Michael Cohen. Here's me. And in fact, we were able to generate this matrix of, if you had a bunch of people and you asked a bunch of people to draw those people, well, we were able to generate this data set. And that allows us to ask really interesting questions like, how would drawer be, which is someone on the internet, as I'll describe, how would they draw me, for example? Okay. So the first question is, how can we collect, we wanted 1 million drawings. How can we collect 1 million drawings with stroke level information? And the answer is we created a game. And basically this game was like draw something, except that you played it with your friends' Facebook photos. So if you and I had a common mutual friend, that friend could be shown to one of us, and then you would start to trace over that person's face. And then the other person would just see your drawing, and they would try to guess as quickly as possible who you were drawing. And I'll show you what that looks like. We also, it turns out, did it with celebrities, and that's because we realized sort of halfway through the project that we needed a lot of drawings of the same face, and so we wanted celebrities are everyone's friend. So there you go, picking Angelina Jolie, and we created this beautiful data set. This game actually launched. We actually did in-game advertising. We literally paid. I think we used NSF money to pay. Like for in-game advertising, we were able to generate about 2,000 images a day while we ran the experiment. Gosh, I'm going to run out of time here. But I want to show you, I think if you watch this video, you'll see exactly what went on. So if you take these images, well, yeah, okay. If you take these images and you were just to run, for example, like a canny edge detector, right? You're not going to get anything that looks like what a human would draw, and it's not very informative. If you average all of the drawings that we got of these people, you actually get these very, very evocative drawings. It's not very useful, though, from a drawing perspective. But it turns out that what you can do is basically create a distance function which says if you were drawing a line at this point, really you meant to be drawing a line there. And so the intuition is basically when you point on the screen, you mean to be pointing to something, and then there's some noise based on where your actual inability to control your finger, and it's the fat finger problem. And so we were able to create this gradient field, and what that allowed us to do was say, if you're drawing Brad Pitt, and you probably mean to be drawing this line, if you're drawing another line, we can figure out a vector that pulls you towards the right thing. And there's a lot more fun math here, how we preserve style and how we figure out if they are not trying to draw some other more stylistic line, but that's the basic intuition. And so here's an example of using this crowdsourced drawing information to very, very subtly modify their drawing. Do you see the lines moving a little bit under the finger? Yeah. So that's the unmodified version, and that's the modified version. And so, and actually it turns out that if, right here this is showing that if you just randomly draw lines, they're not corrected, basically. So we can distinguish between those two cases. Let's see. Let's skip ahead a little bit here. So the point is that this is now a sort of a corrected, the fat finger problem has been solved, drawing of Barack Obama, which actually looks more like what you were trying to draw. And in fact, we did some studies which showed that people couldn't tell that their drawings were being corrected, and yet they uniformly preferred the drawings that had been corrected in this way. How can we tell that they actually preferred the corrected versions? Well, for one thing, they actually undid their strokes less often, and they also, there's a lot more cool results in that study. They also were able to identify the celebrities more quickly, and they were also subjectively rated that they preferred their drawings more. So we're going to run out of time here. So rather than talk about Guess Who, which is an awesome game, I guess I just want to jump to the conclusion, which is well, that I think we live in exciting times, and I think that crowdsourcing really is, I think, this totally still unexplored tool to be able to solve very, very difficult scientific problems, and in particular, extremely complex problems. Also, we were able to identify people like Eli Fisker, who turned out to have sort of like, amazing insight into RNA folding. He's actually a librarian from Denmark, 35-year-old librarian from Denmark, who's just amazingly good at RNA engineering. And we can extract large data sets which were previously inaccessible. So I hope that as you take this back to your own research, you don't just think of crowdsourcing as sort of simple image and pattern recognition tasks, but really as the ability to solve some of the most challenging and interesting problems facing society today. So with that, I'd like to thank my collaborators and thank all of you guys for listening.